Okay, I guess we're on. Okay. Anyway, apologies that this is so late. We were on vacation, sort of, and then it was like I just forgot it's part of it. Okay, so my kids may be running around in the background, which is normal life for me, but you haven't seen that yet. But so this week is when we get into the CSI Crime Scene Investigation Project, and we do the central dogma of biology, which is the whole point of DNA in the nucleus of the cell getting translated and transcribed and then being made into proteins. And then what we can do with that here is sort of the application of that, analyzing it for a crime scene, for a paternity case, all that kind of fun stuff. So genetics review, just an overview of how the structure of a chromosome, the structure of DNA, that kind of stuff. Review, right, from 10th grade biology. Remember that? Or whenever, hopefully. I mean, you were supposed to have biology in high school, I think, but not everybody did, and not everybody remembers and all that stuff. So anyway, there it is. Then we get into the Innocence Project, which is a fantastic organization that tries to exonerate people who've been wrongfully convicted. And no surprise, but guess what gender and ethnic group is primarily the victims of this wrongful incarceration. I don't think I have to tell you that, but it's a really, really cool thing to learn more about and support. Okay, and then we get into a specific case here. So we apply that knowledge to the Watson case. So we can move that picture around a little bit. San Bernardino, and now the really awful thing about this story, it's not these two, they're just a, whatever, a publicly accessible photo. But this actually happened. It was, I think it actually was up around Barstow. Anyway, it says Barstow here, San Bernardino, anyway, whatever. Where, yeah, this is a tragic story of two children found dead in a storage locker. This has been changed, the story's been changed so that it fits this narrative to help you better understand genetics. Papa, I'm recording right now, just so you know. Yeah, just so you know. So recessive, which means, you have two copies of all of your genes, one set from mom, one set from dad. If it's expressed in you, like brown eyes in me, that means that is a dominant trait. If it's hidden, like blue eyes, I probably have a gene for blue eyes, even though my eyes are brown. Both my boys have blue eyes, so that would be a recessive trait, so a hidden trait. So a degenerative disease that's recessive means that the mom wouldn't have the disease, but she could have passed it on to her children. If it's recessive, you have to get that gene from both mom and from dad in order to have the disease. So there's just some details about that, better understanding and application of genetics. And then let's go back to here. So we learn about the Watson case. You learn about a pedigree analysis. So this is a diagram that shows, like, I think circles are women and squares are men, and it shows pairings and then offspring resulting from those pairings. And it's a way to track through the generations specific traits. So if you know that mom and dad both have to have a trait in order for the children to have it, and you know that the children have it and the parents don't, you can you then know that they must both be carriers of that trait, and that it also would have been present in the grandparents. So it's just a way to apply that. Now, what do you do with this DNA analysis? I think I have it right there. Okay, so now this is about the application. So this specifically is a procedure called gel electrophoresis. And the first step is you have a scissors, which is actually an enzyme. Usually it comes from a bacteria that snips DNA always at a specific sequence. So for this, it's GGATCC. Okay, so we know that this is a DNA molecule because it has a thymine in it right there. And it's always going to snip the DNA right at that spot, like right between here. Okay, GGATCC. Now, you got to go look through there. This is just the name of the enzyme, and find these sequences anywhere within that space. So we got GGATCC right there. Cut, it would cut there. And this would leave a segment, 3 segments, 5 base pairs. It's bases, but they pair up, so we call them base pairs. 5 bases long, 11 bases long, and 12 bases long. Nucleotide bases right there. Guanine, adenosine, thymine, and cytosine. Okay? Now, if there's a mutation in one of those sections, it's not going to cut there, and what you end up with is something different. It's up to you to figure out what that different thing would be. Okay? And then you can quiz yourself and see, did you actually do it right? And then we go in for, I think it's here, yes. Just kidding, it's not. a DNA analysis of your case. I'm scared to click that, and then it'll take so long to go back. Anyway, so now we're going to look, and you're going to count. Now this gets a little tedious. I'm sorry, I know it does. I'm trying to remember where it is. GGATCC. There's one for you right there. The next one is, it's like somewhere right around here. GGATCC. There's another one for you. And I know that there's a third one. GGATCC. Did you catch that? Did you catch how much easier I just made that? You're welcome. Okay. Now, if you look down here, what is it? Our GGATCC looks like it has a mutation. Then you go back and you find the other GGATCC and GGATCC, and those are still there. So instead of the number of pieces you'd get here, you'd have less here, right? Because it's not going to snip wherever that, yeah, it's not going to snip here, okay? So you got to go through and count, though. This is like 10, 20, 30, 40, 50 nucleotides. And now when I used to do this on the board, I would actually write in, you know, 60, 70, 80, 90, 100, because otherwise it's hard to get in there and try to count, but do your best, okay? And then indeed, there's a little Quizly. Oh yeah, and I got to go back. It's going to be a pain in the butt, sorry. There we go. Well, that wasn't so bad. A little quiz to see that you actually were able to do it. And then we move on into analyzing the actual gel, which literally is like a square piece of Jell-O. You take a micropipette and you take a little bit of that liquid DNA and you put it onto this piece of Jell-O, and it travels. You put it in an electric machine, very technological terminology there, and you apply a negative charge because DNA is slightly negative. So you got the DNA on this side of the square. The DNA is on this side, the negative charge is on this side, and it makes the DNA repulsed. It moves that way. Although this is the opposite now that I'm looking at this, what I'm thinking of, but either way, it doesn't matter. It moves away from the electric charge and towards the positive charge. The small pieces move faster. So think of it like, what I always use is like the analogy of, what was the guy, Jones, Indiana Jones, where he's got this whole cave full of spider web, right? So if Indiana Jones is trying to get through this and there's just like spider web everywhere, it's just hard to get through there. But you got like a little mouse, right? He takes a mouse out of his pocket and it just like zoom and just runs through there because it's so small. So it can get through a lot faster. So that's the same thing with like a small piece of DNA. It can get through that web, which is truly like a web. A gel matrix is a web. And a large chunky piece of DNA can't get as far. So when you see the gel, I think I have it up here. Do I? No, I don't. Just kidding. You'll see a piece of Jell-O, and it will have like little blue lines, and the blue lines are the aggregate of all the DNA pieces that ended up in that spot. And there's usually like something called a ladder or a standard, and they know that they can use that standard. It's cut into pieces that'll actually like stop at like 10 base pairs, 20 base pairs, 30 base pairs, or something like that. So it's a way to give a relative measure. You don't even need an exact measure, but a relative measure of the ones that got really far down the gel are small pieces, and the ones that didn't get as far are big pieces. And you're just trying to match up someone else's DNA to see if they have the same little blue bands as the sample you're looking at. So for this one, I think we're just going to do it. Yeah, feeling brave. For this one, oh, dude, I hate these. Sorry. You're going to try to see these, whatever this little thing is. Check yourself. There we go. So these are the bands of DNA that would travel. This would be a band that's 5 base pairs long. It's small. And this is a band that's 150. And you're looking at a normal allele. So there's someone who doesn't have that blood clotting respiratory issue would have these bands. And someone who does have the disease would have these bands. But you also have people who might be carriers of the disease that would have this gene and this gene, and then they would have all of these bands in their sample. Anyway. Oh, and we also learned the words heterozygous and homozygous. Just throw everything in there, right? You don't have enough to do already. Homo means the same, and hetero means different. So if you have 2 genetic alleles, 2 genes that are identical, that's homozygous. and two genes that are different, like a normal gene and a disease gene, then you're heterozygous. And then we're moving right along. Okay, so the end of this is actually doing the case report for this Watson case. What specific evidence supports Holly, the mom's, assertion that her children died of a genetic disease? So you want to say what exactly from all this information that we already gathered says whether they did, that they did die of a disease. So you might not agree that they did. You might think that she is guilty of murder. But either way, what of what you just looked at supports her assertion? And then what would be the next steps? If you were the crime scene, the forensic investigator, what kind of evidence would you want to collect? Now, keep in mind that evidence is not really about, there's a word for this, it's not investigate, interrogation. It's not when you question people. That's not really solid evidence. Evidence is the stuff that's done in a lab where there's nobody can lie about it. Okay, so think about evidence as being kind of irrefutable, assuming that everyone's done their job in the lab and all that kind of stuff. Evidence is irrefutable. Interviewing people, you never know if you're getting the real information or not. Okay, and then you're done with that. Oh, by the way, on the 15th, I don't even know what day that is. I'm thinking it. Oh yeah, okay. So this is going to be due on Thursday. This is the stuff that's due by Sunday night. You don't have to wait till then to get working on it. And then we move on into the lab, although it's like lab and lecture because it's kind of all the same now, but introduction to the CSI Project. So we take what we just learned in that lecture content, and now we're going to apply it to a real world crime. The name of the victim changes all the time. So anyway, here we go. Indeed, this used to be a video, but I don't think we get to see the video now. Oh, we do. This is kind of fun. Anyway, plus it has music by The Who, which I love. So learn about the crime scene, the whole process, right? And then we learn about Karen Vinson. She's been killed. This is the case history and interviews with people who were close to her. So you want to jot down some notes as you're reading through this, because it's very important at this point to get as familiar with the case as you can so that it makes sense later on. You don't want to have to be going back and trying to figure out who is this person. So really spend some time now. It will be easy later on. Make it easier anyway. So the autopsy report, I'm trying to talk fast because I don't have a lot of time now. There may be a lot of terminology, a lot of medical terminology. Please understand that there's huge amounts of information here. Spend time to tease apart this content because this is a gold mine of information. And then for the assignment here, refer back to the case file and the autopsy report. A character web, which is something you might do in an English class. How are the individuals related to each other? And you can draw that and take a picture. That's fine. Put it into your PDF or your Word doc. A timeline. Again, you can draw it. There are software programs that will do it too, but drawing works as well. And then based on the autopsy, what abnormalities did you note? Describe them, and then do your own research for real, not that Facebook term. I did my own research, which meant I followed YouTube videos for 6 hours. Cite sources, credible sources here. Proper citations. Go back and remind yourself how to do that. What do you think was the cause of death, and what is the evidence that supports that? Prime suspect and motive, and then how to submit them. There we go, and a little bit of content about how to do so. And I think that's it. Hooray! Huzzah! Okay.